that we've got Rag Aaron from Stanford to talk to us about ultrasound uncaging.
Hey everyone, I recognize I'm the only person keeping you all full on, so I'll keep it brief. Thank you everyone for inviting me for this meeting. It's been really educational for me. I'm a neuroradiologist, kind of playing psychiatry, playing psychiatrist, I guess, while I'm here. So thank you. So I'm going to talk today about a technology developed in my lab for targeted drug delivery using ultrasound. And one of the drugs that we're putting in is ketamine into the system. There we go. All right, I do have some disclosures. The only relevant one is that I'm with Cordance Medical, which is a local to the Bay Area ultrasound company. I'll mention that as I get there. Okay, so why doing this whole ultrasound targeted drug delivery thing? It's essentially this. Currently, status quo for pharmacologic treatments is we give a drug, it goes everywhere, throughout the body, throughout the brain. This is a particular challenge for neuropharmacology, not just because you have exposure to all the non-neuro organs and systems in the body, but also because every few millimeters of the brain, the spinal cord, the peripheral nerves, is functionally distinct. We've heard all sorts of great talks highlighting that here at this meeting. And so, it's not clear that you're going to have the pure on-target effects that you do want from any given drug using that full systemic delivery. And this is, I think, a particular challenge for a drug like ketamine, which is so diverse in terms of all its different functions. It's an analgesic, it's a dissociative anesthetic, it's a treatment for affective disorders, it's also a drug of abuse. And each one of these functions of ketamine is a side effect for the other. Potentially counteractive, potentially goes with it. Not clear if the dissociative aspects can be separated from the affective efficacy. But either way, we would ideally have a way to isolate one function at a time for this. Underlying the complexity of ketamine's effects, even though I'm not in psychiatry, Alan has me contractually obligated to look at the ketamine, opioid link. We did this study in response to the clinical trial results that colleagues in psychiatry did to see whether we can see in a reduced system of a rat some link to the naltrexone pretreatment blocking the effects of ketamine. We used functional ultrasound as an analog of fMRI that we can do in rodents with higher spatial temporal resolution compared to fMRI. And bottom line, we did see changes. With naltrexone pretreatment, we see a whole network of differential effects of the neural activity patterns in the brains of these rats. Generally speaking, the peak was blunted in more frontal brain regions, while the extent of the effect was actually prolonged, particularly in caudal brain regions. Further interesting, though, is we only saw this in the male rats. We didn't see any of this in the female rats. This was an unexpected finding, but a very robust finding. It just fell out of the data. We didn't have to massage it, in order to see this. We weren't looking for these sex-dependent effects. They just were there when we appropriately did the same study in both the male and female rats. Going forward, we then looked at other kind of described bioeffects of ketamine. We looked at PSC95 standing for synaptic density, which has been described as a potential biomarker of the plasticity-inducing effects of ketamine. And we saw analogous results here, where naltrexone essentially eliminated this effect in the male rats and marginally at best affected the female rats. We also saw behavioral effects. So we used locomotor sensitization as a marker of the kind of mesolimbic activation induced by ketamine, and this locomotor sensitization was blocked in the male rats, and basically nothing happened with the female rats. So we have not just an opioid link with the ketamine system and effects, there's a sex dependence. And I'd be really curious to see, I guess, Al mentioned Luke's data, if he sees a kind of similar thing in his imaging. So long story short, ketamine's a complex drug. And we're kind of SOL if we're going to be trying to isolate or optimize one of these effects when the only real levers we have to push are dose and rate of infusion. That said, we at least have hypotheses that each one of these effects seems to be critically linked to action at one or more particular brain regions. And so wouldn't it be great if we could target delivery of ketamine to a critical brain region, and in that way, ideally try to optimize for the intended effect. So that's what we set out to do, and I quickly came to think about using ultrasound as the actuating mechanism for this. That's principally because of this. So many of us are familiar with TMS. I'm not sure if you can see that. Is this better? Okay. So this is the TMS, a current source density map of the TMS field for a typical coil, which is several centimeters wide, spans several cortical gyri. And what this plot doesn't show is that this is only about 4 or 5 millimeters deep. In contrast, this is the ablation zone. Actually, the pointer is wider than the ablation zone. In the left thalamus of a patient who underwent ultrasound thalamotomy. That ablation was done with no skin incision, no craniotomy, and the zone there is about 3 millimeters wide and is in the deepest part of the brain. So ultrasound's really great, physics-wise about sonicating wherever you might want in the brain. But the question is then, how do we use it? Well, what thought to do is if we can make ultrasound sensitive nanoparticulate drug-loaded formulations, a patient would come in, get an IV infusion of these particles, undergo a transcranial focused ultrasound procedure targeting the region of interest. Ultrasound would induce drug release from the particles as they're swimming in the cerebral blood volume of the region, and then that drug would be able to cross the otherwise intact blood-brain barrier, et cetera, in order to mediate its action in the brain. That was at least the vision. With that, we've developed 2 systems of ultrasound-sensitive drug-loaded formulations. One is polymeric perfluorocarbon nanoemulsions, which work for encapsulating and encaging of lipophilic water-insoluble drugs, like propofol, the anesthetic. We've more recently developed acoustic-mechanically activatable liposomes, which works for encapsulating and encaging lipophilic water-soluble drugs, like ketamine. So, of course, I'm going to talk more about the ketamine system. So this is the design of these liposomes. We took a regular plain old liposome, like one that's used for other FDA-approved formulations. And the thing was, how do we make this now ultrasound sensitive? A lot of work went into this, but long story short, you add a little bit of sugar to it, and it makes it ultrasound sensitive. Literally 5% sucrose added to the liposome. That's all you need. And this is basically just some of the data. What we're really doing is we're shifting the acoustic properties of the core of the liposome by adding stuff into it, and 5% sucrose is sufficient to then yield more drug encaging in response to ultrasound. And it's a nice happy medium between potential instability induced by packing more stuff into the liposome in order to get while maintaining the ultrasound sensitivity. So it works in vitro, so then of course, does it work in vivo? For this and for ketamine, we picked 2 different brain regions to look at the encaging in. A medial prefrontal cortical region of interest, it's kind of laggy, and another brain region more posteriorly encompassing retrosplenial cortex and lateral habenula. One thing I'll point out, this looks like a big, huge, beam swath, cutting through the brain. This focus is large for the rat, but it's only a few millimeters wide, and so it's actually relatively going to be targeted when we eventually get to human. And so maintaining the frequency response of the liposomes is intrinsically then determines the size of the focus. So specifically maintaining that frequency response characteristic so that we can have a targeted delivery system for humans. This is not a system really meant for rats. That said, disclaimer-wise, first thing I did is just looked, we used solid phase microextraction to sample the brain that was sonicated or not sonicated to quantify how much ketamine was being delivered. And first thing, we gave just regular old ketamine HCl, the usual unencapsulated free form of ketamine, and saw that both these regions took up and cleared ketamine equivalently. So not only are they interesting in their own right with respect to ketamine, they're nice technical validation regions for this technique and can serve as a control for each other. Okay. So yeah, and these regions are about 7 millimeters apart. So that can give us a sense of the spatial localization too. All right, so with our liposomes, what we see is without ultrasound, or the non-sonicated brain region maintains a similar profile for the ketamine that gets in there. But then with ultrasound, we see a significant rise, effect size of about 2 to 4, depending upon which experiment we're doing. And importantly, the uncaged drug that's added to the system clears from the brain, similar to the free ketamine experiment. And there's a just overall similar profile between each brain region. So great, we can deliver the drug. Does it do anything? So for that, we turn to rat EEG, or ECOG, electrocorticography, very similar to all the EEG talks we just, that nicely teed up the slide before me. So we, first, we recorded from a site closer to the MPFC sonication target or the retrosplenial lateral habenular sonication target. And in response to just free ketamine, so regular old ketamine, what we see is that gamma band. Or we see effects, in delta, theta, alpha, beta bands, but it's really the gamma band that shows us a nice dose-response relationship for free ketamine. Regardless of whether we're looking at, closer to the prefrontal cortex or closer to this retrosplenial habenular region. We'll come back to that in a second. Okay, so then what happens with the uncaging? So what we see is the red traces here are uncaging experiment, whether we're uncaging in the MPFC or the retrosplenial region. And the black trace is dose-matched infusions of free ketamine. So a couple observations. One, in the medial prefrontal cortical region, we see a gamma band induction, like we did with the free ketamine. However, curiously, it's actually somehow more potent and powerful, even though a similar amount of drug is being delivered, and we're just limiting the exposure of the rest of the brain to ketamine. We actually are seeing a little bit more potent gamma band induction here. Further curiously, when we shift the sonication target to the retrosplenial region, while we do see some gamma happening, what we are really seeing is now theta's coming out. Now, if you took a depth electrode and you put it into the brain of a rat or a human receiving a ketamine infusion, what you see is this. You see in the frontal targets more gamma band induction, and you see in these parietal regions more slower waves, so delta theta band inductions. And now we can actually see that in a kind of causal way that local regional ketamine application yields these differential electrophysiologic signatures. And this was all done, not invasively, at least to the brain. Okay, so we can deliver the drug. We see brain regional dependent electrophysiologic effects. Well, what about behavior? So we did this study where we just took the open field test, which is, I'm not a great rodent behavioralist. I'm just going to put that out there. But this is a pretty simple behavior. You have a box, you put the rat in, you use a video camera to record the rat for however long. There's locomotor effects potentially that we can look at here. But also the percent of the time that the animal spends in the center of the field as opposed to the periphery of the field has been described as a marker of just how stressed the animal is. Stressed out rats tend to hug the corners and the sides of the box, while the less stressed rats will hang out in the center of it. So we take these traces that the video camera can see of what the rat's doing and then quantify these different metrics. In this experiment, all the rats got sonication but we varied where the sonication was happening, whether this is at prefrontal cortical target or the retrosplenial habenular target, and also varied what they got, either a saline infusion, free ketamine, or encapsulated ketamine. First, there wasn't an obvious locomotor effect under any of these conditions. They traveled the same distance at the same speed, etc. But with the encaging, when we engage in the medial prefrontal cortex, these animals spent a significant amount more time in that center of the field. These animals had just undergone acute restraint stress just by virtue of the fact that we had to hold them still to, we specifically set up an acute restraint stress paradigm for this, which nicely also allowed us to apply the sonication at the target. And so it seems like that ketamine infusion, specifically in the medial prefrontal cortex, was somehow protective or didn't stress these guys out as much, while all the other animals showed a more usual level of a stress type phenotype. I'm still curious that, though, by limiting in this study, we're not delivering more drug or that much more drug to the medial prefrontal cortex. We're limiting the ketamine exposure in the rest of the brain. So we're actually delivering less overall ketamine to the brain and seeing a more powerful electrophysiologic effect, and in this case, a more powerful behavioral effect. Okay, but that's all rat stuff. This is a clinical conference, and y'all are like, dude, I don't care about rats. Where can we go from here? Okay, so we are working towards clinical translation of the system. At the end of the day, these are liposomes. These are with a little added sugar, so it's eminently translatable. We are fortunate to be funded by the HEAL Initiative to pursue a first-in-human trial of the system. For that, and partly for grantsmanship purposes, because we're targeting the HEAL Initiative, we're applying this to chronic pain as the first application. So the idea is to target ketamine delivery to the anterior cingulate in chronic pain. Already subanesthetic ketamine is being used off-label in this mode of treatment. And the ACC is activated with pain, or intrinsically linked to pathophysiology of pain, to the point that neurosurgeons are doing cingulotomies, ablations of this target for that treatment. And more recently, people are doing DBS of this target for that treatment. So the ACC seems to be where it's at for pain. Also, the ACC lights up when Luke infuses his patients with subanesthetic ketamine. So kind of stands to reason then if we were to target ketamine delivery to the ACC, we can hopefully maximize this therapeutic efficacy. That's at least the hypothesis. Where we are at, we've transferred making the formulation to a qualified CDMO. They've completed the development batches and are in the process of scale up. They could be moving faster. I'm going to go there in 2 weeks to try to get them to move faster, but they're at that point. Then we have to do formal safety tox evaluation. We're in the final contracting stage there, and hopefully the lawyers can just figure it out. For the ultrasound device, we are using that Cordance Medical system. This is that disclosure. I consult with them. But this is what their eventual system will look like. Basically a cap that automatically figures out where it is with respect to the human to apply the sonication to limit operator dependence. We have to make slight modifications to their system for our application, but that's actually the easiest part of this whole thing because it's just their thing. We've had our pre-IND meeting already with the FDA, and we anticipate submitting for the IND and the ID that we'll need by the end of the calendar year to then hopefully start next year. Okay. So with that said, we developed this tool set. The goal is to make a way that we can have a translational tool set so that we can do both forward translation to humans as well as back again to the animals to do these studies, both for focal pharmacologic delivery and also readout assays like the EEG type stuff, the functional imaging type stuff that I mentioned. In a diagnostic mode, could we use this for pharmacologic functional brain mapping to see how these different parts of the brain work in different psychiatric syndromes? Can we predict patient-specific response to pharmacotherapy? On the therapeutic end, can we use uncaging itself as a therapeutic endpoint, or at least as an adjunct to more usual therapies? And on the scientific end, can we at least try to figure out something about how ketamine works in humans? That's probably the part that I'm at least a little bit more excited by. For uncaging itself, so we mentioned the pain example. Well, could we use this at the habenular target, or in subgenual cingulate for depression? What about in pregenual cingulate or amygdala to treat kind of anxiety-related or stress-related syndromes? And for each potential application, how much preclinical data do y'all need before I ring you up to do a clinical trial of this? So with that, thank you, and yeah.
Well, I think that is bang on time. Thank you very much indeed. I'm afraid we don't have time for questions. We've got a very tight lunch break, but with the message that a spoonful of sugar helps the ketamine go down, I think that's a good point to end on. So thank you very much indeed. It's wonderful.